I love it when she says that. And once again, I'd like to welcome everyone to the Los Angeles Birders webinar for November 12th. We have a wonderful program coming up. But if you're not a member of Los Angeles Birders, please consider it. Your small donation, a yearly donation of $20, $10 if you're a full-time student, includes hosting and recording and storing these webinars that you're about to see, includes helping out with field trips. We have lots of educational opportunities such as Birding 101 and 201. Many out there are 101 and 201 graduates. Welcome, everyone. We have lots and lots of student learning opportunities, including scholarships to help our young birders, help our students do what they have to do. And so all of your, and we have lots of science opportunities. And so all of your contributions really help out with those things. And with that, I would like to introduce our community science chair, Lance Benner, to introduce our program for tonight. Lance?
All right. Thank you, Ron. Yeah, tonight's speaker is Dr. Devon DeRaad from UCLA. So the title of his talk is going to be Identifying the Source and Potential Spread of Introduced White-eyes in North America. So many of you may have met Devon when he was an undergraduate at Occidental College several years ago. He made many contributions to our understanding of the distribution and status of birds in the Los Angeles area through many eBird reports that he contributed when he was at Oxy. And he also started doing research at the Moore Lab using some of the specimens there while he was a student. So he then went on to the University of Kansas, where he earned a PhD this year, earlier in 2024. And so he's now an evolutionary biologist and a postdoctoral researcher at UCLA. So he's involved in doing a number of projects, including some work on scrub jays here in California. Tonight's topic, however, as I mentioned briefly a moment ago, is going to be trying to understand the sources and spread of the different white-eyes that have been found in the United States. And so with that, please welcome Devon DeRaad as our speaker tonight. Take it away, Devon.
Awesome. Give me a second. More my slideshow, please. How does that look?
Great.
Oh, there, you're perfect now.
Awesome. Okay, thanks so much, Lance, for that really fabulous introduction, and thanks for having me here tonight. I'm excited to be talking to you about some of the work that I have sort of been thinking about for a long time, and I'm hoping to continue as I keep developing my research program focused on the white-eyes in North America and elsewhere. I want to start by acknowledging that the work that I'm going to be talking about tonight is really collaborative and really relies on a ton of museum specimens and a ton of open collaboration from all of the museums shown here. And this work just simply wouldn't have been possible without, that collaboration. And as I mentioned, there's not just museums, but actually people who work in those museums who provided the specimens, provided feedback, provided help with analyses, provided help with writing. All of the people here were integral in bringing the work that I'm going to be presenting to you tonight together. And I did want to mention that the bulk of the stuff I'll be talking about tonight is part of a published paper. It was published in Biological Invasions earlier this year, and if you're interested, I would encourage you to go read that paper for all the nitty-gritty details. Okay, so this is an outline of what I'm going to try to go over tonight. I'm going to start by talking about how Zosterops, or the white-eyes, are known as the ultimate great speciators, and then I'm going to talk about the white-eyes in Southern California in particular, how we use genetics to study them. I'm going to talk a little bit about more on the ecology side, how we're using environmental data to potentially forecast the spread of these birds. And I'm going to talk about future directions of where I think this exciting, at least I hope you find it exciting, research is going. So this is like the typical EEB slide that every ecology and evolutionary biology talk starts with, which is like a picture of tons of pretty organisms. And the goal of almost every ecology and evolutionary biology researcher is to understand how evolution has shaped the diversity that we see today. And I would argue that birds, I probably don't have to convince you that birds are the best way to study that because they have this incredible diversity. And you see all of these gorgeous rainbow of colors in these birds. So you might be thinking, why study white eyes, which don't exactly have a gorgeous rainbow of colors, but instead are just this sort of gray and green and yellow phenotype in general? And the answer, in my opinion, is because they are a great speciator. And to put that in context, what I mean by a great speciator, I wanted to talk about a group of birds that you might be a little bit more familiar with, which is the Psittophaga warblers. This is a genus of warblers that is in the New World. You've probably seen a bunch of these birds. They're considered, incredibly diverse. They've really rapidly radiated, and they have all these different species. But in about 5 million years, this genus has accumulated about 34 species. When we compare that to Zosterops, which is the genus that contains all of the white eyes, in just about 2 million years, Zosterops has accumulated 111 species. So this is about a 5 to 10 times higher speciation rate in Zosterops than something that we might consider a pretty speciose genus in our part of the world. And actually, there's evidence at this point that Zosterops are one of the fastest diversifying vertebrate lineages. This is a paper from 2009 that was actually led by my grad school advisor, Rob Moyle. And it talks about how Zosterops are these great speciators and uses genetic data to identify the genetic diversity in the group and compares them actually to the cichlids from the African Rift Lakes that are known as like some of the most incredibly diverse and rapidly speciating organisms, at least vertebrate organisms on Earth. So a main question is, how has Zosterops, these white eyes, diversified so rapidly? And you can see in this tree that was published recently, earlier this year, that there's this big burst of diversification in white eyes about 2 million years ago, which is shown in that red box with the arrow there. And this burst corresponds with rapid island colonization. So the white eyes are known for their ability to disperse, to leave wherever they're at, get up, fly to a new island, and once they get to that island, to colonize it successfully, establish a new population, and to differentiate really rapidly and actually become new lineages, subspecies, and species on all of these tiny islands throughout their native range. So Zosterops are great speciators, as I've established. They're incredibly diverse, despite not being incredibly phenotypically diverse. And you might still be asking, why does that matter? And I'm going to talk to you a little bit about the speciation process, because to me, Zosterops is so important because we need natural model systems for studying the speciation process. And I want to start by going over our current understanding of the speciation process and how speciation happens in birds. So typically we start with one species down on the bottom of this cartoon diagram here, and then that species splits. And for a while, the two isolated lineages are in this speciation gray zone. And it's not really clear whether the two species are on independent trajectories or whether they might just come back together. And finally, up here, the two species are so divergent that they're completely isolated. And I would argue that we have a pretty good understanding of this speciation continuum at this point. Starting from studies of single populations, like the classic studies by Nancy Chen and others on the dynamics of the pedigreed and intensively studied Florida scrub jay population in Central Florida, to the studies of warbler species that have hybrid zones and come back together in secondary contact and hybridize, like the Townsend's and hermit warblers. There's tons of awesome work on these birds from Silu Wang. And another example is the northern flickers, where my postdoc advisor, Stephanie Aguillon, has worked on the genetics of their entire distribution, as well as the hybrids in the Great Plains and the genetics of the color differences between these two birds. And all the way up to the sort of top of this diagram, where there's been a number of studies on the landscape of genetic divergence between isolated species that are reproductively isolated and very divergent, and two good species, like, for example, Anna's hummingbird and Costa's hummingbird. But I would argue that there's a part of the speciation continuum that we're largely missing. And that part is like right sort of at the base of this gray zone. And this is a challenging area to study, especially for speciation studies. And the reason I think is shown by this cartoon diagram. So you see here that we have at the bottom single population studies where all of the cartoon butterflies are white and are the same color. And then we have these speciation studies where there's two different things on each side of the river, and they're isolated, and we're like, wow, some butterflies are white and some are purple, and we need to study the differences between them. But this zone right here, I'm calling it the too early to tell zone, where the white butterflies are separated by the river, but we don't really know what's going to happen evolutionarily. It's too early to know if these two butterflies are on independent evolutionary trajectories and going to turn into multiple species and should be studied for speciation, or whether that river is simply going to evaporate or dry up and the butterflies will come right back into secondary contact and swamp and become a single population again. So speciation studies at the top focus on how population divergence is maintained, while this too early to tell zone is much more difficult to study. And I'm going to argue that the great speciators can be used to study this too early to tell stage of the speciation process. And I think Zosterops is ideal for this because it seems like every time the white-eyes reach a new island, they become a new species. So if we can find newly founded Zosterops populations, this would be an ideal chance to observe the rarely studied early stages of the speciation process. Enter the Zosterops in Southern California. So I did my PhD at the University of Kansas, studying under curator Rob Moyle, as I mentioned earlier. And he has sequenced and studied Zosterops populations from across the globe. So my context for white-eyes during grad school was their awesome speciation ability in their native ranges. But before moving to Kansas to do my PhD, I was an undergrad at Occidental College in Northeast LA. And I highlighted Occidental College with a little red star on this map. And as early as 2016, I was aware that there was a population of white-eyes in Orange County. And at the time, these were just a cool new bird to me that I hadn't seen before. So I was willing to drive down to Orange County to get to see them. And here's my eBird checklist and photo from December 2016, where I went with a friend and saw a flock of 12 white-eyes in, I think it was Newport Beach. And this has actually been changed in the intervening time period, but one of the things that stuck with me when I made this checklist in 2016 was that the population was identified at that point as Japanese/Swinhoe's white-eye. And I think the reason that the identity of this introduced population was in doubt at the time should be clear from what I mentioned earlier, that the genus includes 111 species, and all of them essentially look something like this, with some subtle variation of this yellow, green, and gray warblery phenotype. And the reason that these two species in particular it was unclear whether they had founded this Southern California population, is that they not only look extremely similar, but they're also some of the most common species in the global songbird trade. And there's even more Zosterops species in the global songbird trade. This traffic report from 2017 did a survey of songbirds in Singapore pet shops, and they found that actually Zosterops palpebrosus, so not Japonicus or simplex, was overwhelmingly the most common bird identified in the pet shops of Singapore. So at this point, I was like, well, there's at least 3 possible species that could have founded this introduced population in Southern California. All of them are extremely challenging to identify by appearance alone. You can see they look incredibly similar. And there's yet another, even more challenging possibility, which is that we think the Zosterops population in Southern California came from the pet trade, where many different white-eye species that are challenging to identify correctly are routinely kept together, creating the opportunity for hybridization and even more complicated genetic ancestries. So in order to resolve this complicated, potentially complicated history, I wanted to see if I could use genetics to identify the origin of the Southern California white-eye population. And as I mentioned earlier, when I became interested in this project, I was doing my PhD at the KU Natural History Museum in Lawrence, Kansas, which has one of the best collections of tissue samples from wild Zosterops populations of anywhere in the world. So I decided that I was the right person to go and answer this question. So I used this exceptional tissue collection along with lots of loan specimens from other museum collections and collaborators to generate a genetic data set from 6 possible Zosterops species that could have plausibly been involved in the founding of the Southern California population, starting with Zosterops erythropleuris, which I'm showing here, which is a migratory species that breeds at high latitudes in Russia and northern China. And then I also included Zosterops everetti, which is this species shown on the slide that is endemic to the southern Philippines. Zosterops japonicus, which is widely distributed from Japan all the way down through Indonesia. Zosterops nigrorum, which is endemic to the northern Philippines. Zosterops palpebrosus. This is, if you remember, the Indian white-eye, which was the most commonly found in Singapore pet shops. And Zosterops simplex, which is found throughout China and into Southeast Asia. And in addition to all of these 6 native range species, I also obviously wanted to use those as a reference to identify the goal of this whole project, which was to identify the source of introduced Zosterops populations. So in order to do that, I had also former Oxy undergrad Madhvi Venkatraman send me white-eye samples from Hawaii. She did a PhD at the Smithsonian where she worked on the Zosterops population on Hawaii. So she shared tissue samples with me from that population. And Allison Schultz, the curator at LA County Natural History Museum, who had 3 samples from the Zosterops population in Southern California that we could use for genetic analysis. And the approach I used is called RADseq. It stands for Restriction Associated DNA Sequencing, Restriction Associated Digest, something. I messed it up, but RADseq, the idea is we're using a restriction enzyme to cut the genome at a bunch of different places and get a bunch of different pieces of DNA from across the entire genome that gives us a representative sample. And we can then align those back to a reference genome for the species, and we get thousands of pieces of DNA from across the whole genome that allow us to reconstruct the evolutionary relatedness between all of the samples we've sequenced with really good power, because we have the statistical power of thousands of individual pieces of DNA from throughout the entire genome. And our genomic data allowed us to reconstruct the genome-wide relatedness among all of these samples from these 6 Zosterops species that I mentioned earlier. And this is an example of a phylogenetic network that I built from this genomic data. And the way to read this network is that the distance between 2 samples corresponds directly to how genetically divergent they are. So if we find 2 samples like this one and this one, they're genetically closely related because we only have to track a small distance through the network in order to connect them. In contrast, if we wanted to connect samples from the simplex species up here with Japonicus, we have to travel a large distance through the network in order to connect these samples, which just means they're more genetically different. So now that you know how to read the network, what should stand out is that the 6 species we sequenced form these 6 really discrete genetic clusters. And those correspond again to simplex, everdi, nigrorum, palpebrosus, erythroplurus, and japonicus. And the fact that these 6 populations were really tightly clustered is exactly what we expect from 6 different species. Except for these 2 samples, which actually don't cluster with any of the 6 species and show up sort of in the middle of this network. And we think that these birds are likely hybrids. And that's because they're essentially right in the middle, where if you trace through the network to try to understand how genetically related they are, they're equally genetically related to Japonicus, this sample, and to simplex, which is indicative of a hybrid ancestry. And when we look on our map, those 2 hybrid birds came from localities where there's more than one Zosterops species known. So it makes a lot of sense that these are areas of contact between multiple species where hybrids might show up in our data set. So we've identified the 6 species, we've identified the hybrids. The next question is, where are the introduced Zosterops populations coming from? And this Hawaii introduced population in particular is shown by the white dots in this cluster over here within Zosterops japonicus. And you can see that the Hawaii introduced population is deeply nested within this Zosterops japonicus clade. And that means that we think they are pure Japanese white eyes. They're not hybrids or any other species. And in contrast, the white eyes from Southern California are nested deeply within this Zosterops simplex clade, meaning that we're confident that the birds in Southern California are swinhoe's white eyes and not some type of hybrids or any other species based on our genome-wide data. And it's important to note that neither introduced population appears to have a hybrid origin, which was one of the main original hypotheses for me. Is I thought maybe coming from the pet trade, they would be some weird mix of multiple species, but neither the Hawaii population or the Southern California population shows any evidence for that. So now that we've identified the species that founded each of these populations, I wanted to dive a little bit deeper with our genetic data and ask whether we could identify the exact source of the introduced populations, specifically starting with the Zosterops japonicus, or Japanese white eye, on Hawaii. So I made another map here and I labeled all of the individual sampling sites with numbers just to keep them straight. And this is a principal components analysis. It's sort of similar to the network that I just showed in the sense that each dot represents an individual sample, a genetic sample in our data set, and the distance between samples is telling us how closely or distantly related those samples are to one another. So all of these samples from populations 15 and 16, which is these 2 very geographically nearby islands, are really closely related to each other, but they're distantly related from all of these samples that come from Japan and the Korean Peninsula. And the important thing to note here is that the Hawaii samples, these white dots, which are population 1, are actually nested really closely to other dots, other genetic samples from population 2, 3, 4, and 5. And 2, 3, 4, and 5 are from Japan and the Korean Peninsula. So this is suggesting to us that the population on Hawaii is really genetically almost indistinguishable from birds that are on Japan in the wild today, which is really cool. And another question I had was, what would the genetic diversity look like in these introduced populations compared to their native ranges? So now at the bottom, I've laid on this plot of genetic diversity where the y-axis is showing heterozygosity and pi, which are just measures of genetic diversity. And what we see is that this Hawaiian population of Japanese white eyes is actually at or near the top of the amount of genetic diversity from all of the populations that we sampled, with some of these tiny island populations like island 20, 21, and 22 showing reduced genetic diversity, which makes sense. But these Japanese white eyes on Hawaii don't seem to have suffered any sort of genetic bottleneck or reduction in genetic diversity following their translocation to Hawaii. So I wanted to take the exact same approach for the Southern California population to see if we could understand what was going on in that population as well. And I did the same thing here. I made a map and labeled all of my sampling localities with numbers. And for the swinhoe's white-eyes in Southern California, you can see here in the principal component analysis that unlike the Japanese white-eyes, the introduced population from Southern California over here is really genetically distinct. It's on its own. It's not close physically to any of these other samples. And the thing to note here is that we have this big sampling gap. So we have a lot of samples from China, and then we have one sample from the bottom of Southeast Asia over here. And in between, we're missing samples here. So it's possible that the Southern California birds come from a region that we have not sampled, and that if we added more native sampling, we would be able to know the exact location that these Southern California birds were translocated from. But it's also possible that these Southern California birds are already genetically diverging from their native counterparts, or that they genetically diverged during time in the pet trade and that they've become adapted to the pet trade. And I wanted to do the same exact thing that I mentioned for the Japanese white-eyes, which is compare the genetic diversity in this introduced population to all of the native populations. And what you can see here, this gray asterisk that's behind the dots, is actually pi, or overall genetic diversity for the population. And the pi level for the Southern California population is actually the lowest of any swinhoe's white-eye population that we sequenced. So this is suggesting that unlike the Japanese white-eyes in Hawaii, the swinhoe's white-eyes in Southern California are actually really genetically bottlenecked and have lower genetic diversity than any of the native populations that we sequenced. So another major question that I had was, why are the genetic dynamics so different between the introduced populations in Hawaii and Southern California? Again, the ones on Hawaii are closely related to source populations in Japan and Korea. They have comparable levels of genetic diversity. Meanwhile, the swinhoe's white-eyes in Southern California are not related closely to any of the populations we sequenced from their native range, and they have this genetic bottleneck. I think the main explanation for these differences in genetic dynamics is the huge differences in the founding dynamics between these populations. So as part of writing this manuscript, I was doing some digging in the literature, and I found a paper that specifically says that 252 Japanese white-eyes were intentionally introduced to Hawaii in 1937 for the purpose of mosquito control and insect control. And I think that massive influx of birds, that huge founding population size, essentially subsampled enough of the genetic diversity in Japanese white-eyes that it allowed them to avoid any sort of population bottleneck and allowed them to basically very quickly expand. Meanwhile, the swinhoe's white-eyes in Southern California, only 4 birds were observed in Newport Beach in 2006. That's the first eBird record. And no group of greater than 4 individuals was subsequently observed in Southern California for over 5 years, which suggests that this population was potentially simmering at a really small population size and inbreeding a lot, creating this genetic bottleneck before it started expanding. And maybe an obvious next question is, how will this genetic bottleneck in these different histories affect the trajectory of the swinhoe's white-eyes in Southern California? And obviously the Japanese white-eyes had no genetic bottleneck that we can detect, and they ended up going on to become the most numerous bird in the Hawaiian Islands. They're incredibly successful. So I was curious, because we find evidence for a strong genetic bottleneck in the swinhoe's white-eyes in Southern California, is this going to lead to inbreeding depression and eventual population collapse? And this might not be a surprise to a lot of you, but I'm going to put the maps up in order just to show you whether, or you can decide for yourself whether or not that's true. This is the distribution of Zosterops simplex in 2012. There was population in Orange County, and a single observation from, not sure what that area is, north of Santa Monica, maybe Westwood. 2014-15, there was tiny expansion, not much. 2016-17, you start seeing big geographic expansion in this white-eye population. 2018-19, you get even more white-eye expansion. You get the first observation on Catalina Island and the population popping up in San Diego. By 20 and 21, this population in San Diego is expanding as well, and there's now another island colonized down here. The population's expanding extremely rapidly. These are figures from the paper, so they're actually already out of date, but the 2022-2023 map is showing that even just in recent time periods, there's been a ton of white-eye sightings. The red bubbles are obviously birding hotspots from the last month, if you're familiar with looking at the eBird range maps. So I had someone ask me when I gave a previous version of this talk, could this just be the fact that more people are birding now, more people are putting eBird checklists in, and they actually haven't increased, but we're just detecting them more. And maybe I don't need to explain this to all of you, but Southern California is one of the most heavily birded areas in the country. And I promise you, people were birding a lot in 2012 to 2013 all over Southern California. And if white eyes were out there, they would have been detecting them. I feel pretty confident that this is a real signature of geographic expansion, really rapid geographic expansion, especially over the past 10 years. And that's what this slide is about, is that this population has really exploded in less than 10 years, going from very geographically restricted in Orange County to sort of all over the LA basin, although not making it much all the way up to Pasadena. It's sort of hugging the coast, but definitely expanding up and down the coast, now spanning 400 miles of coastal Southern California. I did this slide today where I looked up how far is it from Ensenada to Morro Bay, and it's over 400 miles. You can see now the southernmost observations are in Ensenada, Baja, and Morro Bay is the northern extent of the distribution at this point. So how will this bottleneck affect the trajectory of simplex in Southern California? We can cross out that hypothesis that inbreeding, depression, and population collapse are going to set in. And it seems that we can answer the question of whether strong genetic bottlenecks are going to limit the spread with a resounding no. So the next question was, what is going to end up limiting this white-eye population if it's not genetic dynamics? And for that portion, I'm going to be talking about projecting the spread of Zosterops simplex using environmental data. And to do this, I partnered with Marlon Kobos, who was a fellow graduate student at the time working at the KU Natural History Museum. And his research focuses on distributional ecology and ecological niche modeling. So I was focused on the genetic side. I said, I need someone who knows what they're doing on the niche modeling side. Pitched the project to him and asked him to build some niche models for me. And what he did here is use the distributional information from the native range of Swinhoe's white-eye, which we're now confident is the population that founded the Southern California white-eyes. And he generated an ecological niche model, which shows the areas with the most suitable combinations of environmental conditions in these red hot colors on the heat map that's shown on the left here. So Swinhoe's white-eye is throughout this distribution, and really the core of the distribution is here, and the environmental conditions of what looks like southern China are really its ideal conditions according to our analysis and our model. And we were able to then plot those suitable conditions onto a map of North America to show us where the ideal environmental conditions for these birds are found. And it's pretty much exactly what we expected, which is that subtropical areas like coastal Mexico and the southeastern US would provide highly suitable habitats for Swinhoe's white-eye because they match the subtropical habitats of the species' native range. But when we zoomed in on the area in Southern California where the introduced population is currently residing, we found that the current distribution was projected to have only low to moderate suitability based on the conditions found at the native range. And you can see this is, again, outdated, but this was at the time the extent of the population in Southern California shown in these dashed lines. And you can see these blue colors and like light yellow is indicating that this habitat is not supposed to be terribly suitable for Swinhoe's white-eyes. And this could mean multiple things. It could mean that the novel ecological conditions of Southern California will eventually limit these white-eyes, and that the population will contract and they won't be able to establish and live long-term in Southern California. I don't think that seems terribly likely based on what we're seeing from the eBird maps and the fact that they're constantly expanding, and they're certainly breeding and very established in Southern California. And I think the second more likely explanation for this surprising result is that there are factors in our ecological niche model, factors that our ecological niche model is not taking into account that are allowing these swinhoes white-eyes to actually succeed in Southern California. And some examples of those could be ornamental fruit trees that they're using as food resources, urban heat islands that are allowing basically warm microclimates that help them get through the winters, all kinds of anthropogenic urbanized resources that these birds could be exploiting that aren't included in our niche model. And if that's the case, then our niche model may be overly conservative and may not recognize that there's a bunch of appropriate habitat up and down the coast. And if that hypothesis ends up being true, I would expect to see the swinhoes white-eyes continue expanding, especially along urbanized corridors and between cities. And ultimately, only time will tell how far these birds are able to spread throughout North America. Okay, so I'm going to end on some future directions and talk about why I think it will be important to continue studying the swinhoes white-eyes of Southern California. I mentioned early in the talk that this too early to tell zone in speciation studies is very hard to study because it's hard to know which populations to study in order to actually observe speciation in action, because we can't predict the future and we don't know whether two populations that are isolated are actually going to turn into two species or not. And I'm going to argue that this population of white-eyes in Southern California offers an ideal look into the early stages of the diversification process. And we can actually view the translocation of swinhoes white-eyes to Southern California as a natural experiment where humans translocated these birds from their native range into a new, completely isolated region. Despite how strong of dispersers these birds are, there's no evidence that any of the birds from the native range can make it all the way across the Pacific and all the way to California on their own. So we know this population in Southern California is completely isolated, and we can now study this experimentally translocated population to observe the process of divergence in real time in a completely isolated population. And an even cooler aspect of the system is that not only is this population in Southern California isolated, but the colonization of islands and the divergence process within the Southern California population is playing out again in real time, as these exceptional dispersers have now colonized from the mainland onto Catalina and San Clemente Island. And this is just in the past few years. And if you remember back to the beginning of the talk, I spent so much time setting up that Zosterops are known as great speciators because they have this proclivity for colonizing new islands and rapidly diversifying. And I'm arguing here that this introduced population in Southern California might be in the process of repeating this explosive evolutionary trajectory in a totally new location this time in Southern California and on a new set of islands. And we have the opportunity to study that process as it happens in real time. And identifying tractable experimental frameworks for observing this rarely studied too early to tell zone in speciation, I think will allow us to ask some really exciting questions that most speciation studies that are performed at the later stages in the process can't address. And those major questions include, how do novel populations overcome the invasion paradox of low founding genetic diversity, which I documented in the swinhoes white-eyes of Southern California? How rapidly can isolated populations diverge from their sources? What are the relative contributions of natural selection and genetic drift in creating divergence in newly isolated populations? And how does the genome respond to the isolation process, essentially? What is the landscape of genetic divergence? And are there specific features of the genome, like sex chromosomes or chromosomal inversions, that are more rapidly diverging than other parts of the genome. I think this is a really understudied aspect of the early part of the speciation process and something we might be able to get at with this really cool system. So thanks for your time. I'm really happy to take questions. I think we've got a couple minutes, and I wanted to end by saying, please get in contact with me and or Allison Schultz, LA County Museum Curator, if you ever find a dead or injured Zosterops. I've been told that bird rehabbers in Southern California won't take Zosterops because they're not native. So we can't get them from bird rehabbers, but if you find one that's hit a window or been killed by a cat or something like that, it would be awesome to get it to a museum collection so that we can use that unfortunate event as an opportunity to study these birds and learn more about their DNA and stuff like that. So
Absolutely, absolutely wonderful. That was a wonderful talk. If you have any questions for Devon, please pop them into the Q&A down at the bottom of your screen. It kind of
Yeah,
helps.
you might have to click the more thing to get the Q&A depending on your
Yeah, if you don't have a
Zoom
Q&A.
version. If you don't see a Q&A at the bottom.
Devon, that was
yeah,
fascinating.
fantastic.
I really, really enjoyed that.
Yeah, it was. I have one question about, you showed an eBird map. I think it was the last one that you showed, that there seemed to be a gap between Orange County and San Diego where there were no white eyes. What's going on with that gap there? Were there two populations, or just a region that, there? Yeah, there seems to be this gap.
that's a great question. My interpretation is that this is Camp Pendleton here. I think if you've driven from LA to San Diego, there's this area that's like an Air
Right.
Force base. I'm not sure if it's still active. I think
It
people
is.
are not really birding here. And if you look at the maps sequentially, it really looks like, right, they were moving down, moving down, moving down, and then it looks like they made this big jump. But my interpretation of that is that that's just an area that's not birded.
Okay.
Continued down to San Diego. So I think this is a single founding from that Orange County population that has now jumped down to San Diego and is going basically north and south up the coast at the same time.
Okay, cool. Thank you.
Yeah, Mark, we have some questions.
We do.
Oh, and Devon, we're going to, since people on the recording won't be able to see the questions, we're going to actually read them out to you, if you'd
Perfect.
like.
Yeah. So we have a question from Mark saying, asking, what time frame might you expect the white eyes to evolve into a new species in California?
That's a great question. To pull up the phylogeny again. This is the thing. I mean, normally we think of the speciation process as so slow, right? We think of allopatric speciation as being, species have to spend millions of years in isolation, just slowly drifting to become new species. But Zosterops is one of the prime examples that vertebrate speciation can be really rapid. So that's basically one of the outstanding questions, is like, how fast will we see genetic divergence between this Southern California population and other populations? And I think a lot of that might be mediated by the strength of the genetic bottleneck. It's not really well understood how genetic bottlenecks essentially interplay with the divergence in speciation process. And it's also dependent on the degree of natural selection. I think if there's really different environment in Southern California that for whatever reason is selecting for some extremely new phenotype and new sets of alleles, we might see really rapid divergence. So it's a great question, and that's what I want to keep studying for sure.
Yeah.
Hope asks, has anyone studied the calls and sounds of the Asian population and our population to see if there's any differences in vocalizations?
That's a good question, and that's, not my expertise, unfortunately. I think that's my weakest area. I think birders would tell you they knew these were swin-nose white eyes long before I did with genetics because they studied the vocalizations. But I don't have a good handle on whether they're different in Southern California. or how different they are across their range throughout sort of China and Southeast Asia as well.
But you're working on a grant to send you over there, correct?
I wish that would be great.
Cool.
It sounds like it would be useful if people obtain recordings when they find the birds and include those recordings in their eBird lists, so that
Yes.
this sort of thing can be investigated by people like you as the sample sizes grow and the geographic distribution and the expands and time elapses.
Yes, we should
Excellent
tell
point.
people in southern China to do the same thing with their birds.
Exactly. I see a question in the webinar chat that says, are you seeing adverse effects to other species that they're competing with in Orange County, thinking of bush tit populations?
Oh,
That's a good question. I actually, like, in some of the background research for this manuscript, found that there was a population of, I think it was Zosterops palpebrosus, the Indian white-eye, that got out of the San Diego Zoo in the 70s or 80s. And they actually, I don't know, like Fish and Game or someone who's in charge of that, basically went and exterminated the birds because they were worried they were going to turn into an introduced feral population. And they were saying that they cited, I think, fruit like crops in Southern California as,
yeah.
that these white eyes could decimate fruit crops in Southern California, and that they were a big concern as an introduced species, which I thought was really interesting considering that now they're everywhere and nobody seems to care. So I'm not sure what's changed in
Well,
our
a lot of attitude has changed. Same thing happened, of course, with the bulbuls in Southern California.
Yeah, so it's a good question. If you noticed throughout the presentation, I tried to refer to them as introduced rather than invasive. My understanding is that, like, they're translocated, they're non-native, they're introduced, but invasive implies that they're sort of, outcompeting other native populations or having an adverse effect. And we don't have any evidence that they're doing that at this point. But that doesn't mean they're not. It just would be really hard to quantify.
Right, right. Interesting.
Great.
Mark?
Tori asked a question. Are there any plans to explore the geographic areas in Asia that don't have much data that might clear up the origin question?
It's a very good question. The short answer is yes, I would love to add genetic samples. It's just a question of finding the right collections and getting the right samples. So I worked with the samples I could get from KU and my collaborators, but yeah, I would love to fill that distribution in more so that we could be really confident the way we are with the Japanese white eyes in Hawaii, where we can almost pinpoint exactly that these birds were introduced from mainland Japan. With the white eyes, it's much more, with the Swinhoe's white eyes, it's much more unclear. And I agree, adding sampling would help clarify that a lot.
John asks, it looks like there are now sightings from around 20 different locations in the San Francisco Bay Area. Do you know
No.
available specimen material? Any thoughts about whether this is an independent introduction?
That's a great question. I mentioned, might have been before we started broadcasting, that there's also like 1 or 2 sightings from the Seattle area, which is even further up. And I showed this map. Yeah, I thought someone might ask this question because this map actually shows all the way up in Seattle, like that they're really up and down the coast. My understanding is that all of the populations north of Morro Bay are independent pet releases, that a handful of birds in the bay and even further north have been released, but that there's no evidence of them breeding and establishing populations, and that those are totally independent from the Southern California population. And it's really interesting to study invasive species because they do seem to generally follow this sort of explosive trajectory that the Swinhoe's white eyes are following, where for years there was what seemed like a handful, a dozen Swinhoe's white eyes in Orange County, and they weren't expanding. The population didn't seem to be getting bigger. It was just sort of this little novelty. And then something switched, a switch flipped, and suddenly they can just go crazy and expand. And long term, what often happens with birds like this is that we've seen this with like European starlings and other birds, that they'll expand like crazy, and then something will again flip back and the population start to shrink. And we don't have a great understanding of what the details of those dynamics are and why populations tend to follow those trajectories. So I would love to keep studying invasive species to try to understand that better.
You mean introduced species?
Yes.
Yes.
Thank you.
Great, thank you. Kimball asks, it's basically a follow-up question to this. Do you think there's likely that there are continuing releases of white-eyes, swinhoes, or others into Southern California from the pet trade, and how that might affect population genetics?
That's a great question. Yes, the answer is yes. I think reading that the traffic report that I cited here talked about that there's a tradition among some religions of, I'm blanking on what it's called it, merit releases, I think it's called, that basically by setting the songbirds free, you are, being a good person, and it's part of the religion. And so I think,
And also marriage ceremonies, things like that, I think.
yeah, I mean, I think there are certainly maybe just pets getting free, but also potentially people letting stuff go on purpose. And I think that's definitely affecting the population genetics. It makes it harder to study, unfortunately, because it's hard to know if you go out and find a white eye. Is this a bird that has been adapting to Southern California for 15 years, or is this a bird that got out of someone's cage yesterday? So I don't actually, it's a good question, Kimball, but I don't like to think about it that much because it makes studying it even harder. But the thing that I fall back on is that we can look at the population trajectory. If I go back to those maps, right? And like going the wrong way, sorry. If we look at the population trajectory, we know that as recently as 2012, something like that, there was only a handful of swinhoes in Southern California, and today there's thousands and thousands. So by that logic, an overwhelming majority of the birds that you see in Southern California should be born in the wild in Southern California via this population expansion. So we can be relatively confident that most birds we see are now native to Southern California and adapting to Southern California, which is pretty cool.
That's cool.
That's great. Question from Yvonne. I came across a 1984 paper from University of Nebraska, Lincoln, about the white eyes eradication effort in California. Were any survivors from this population completely separate from the population you studied?
That's a good question. I assume that would be the San Diego Zoo escapees that I mentioned earlier. And I think they were successful in eradicating that population. I think it was just a handful of birds that got out from the San Diego Zoo and they were able to catch them all. I don't know how, but yeah, I think that's completely separate from this introduction that we've seen.
Lily asks, do you think that the white eyes are invasive?
In any way, yeah.
Yeah, it's a good question. I mean, someone mentioned that they act similar to bush tits. It certainly seems like they could outcompete bush tits. But I'm sort of hoping that they're more like the parrots in LA, that they specialize on anthropogenic food resources, right? Like introduce ornamental trees and fruits and stuff like that, and that I mean, sort of like house sparrows too, right? Where house sparrows' main diet seems to be like French fries and stuff like that.
Burger King, yes.
Right. They're not actually interacting and directly out competing native birds all that much, but it's hard to say. Ecology is really challenging. Understanding what's happening across a big landscape like Southern California would take, unbelievable amounts of effort. So I feel lucky that I can do the genetic work and get a ton of data without having to leave my lab, which is pretty awesome.
Yeah, that's fun.
Very cool.
Don't get as much birding done, but it's not bad.
It's true.
Yvonne asks, disregard my previous question. Oh, okay. The study indicated they were omnivorous.
Huh? Really?
I'm sure they're feeding their babies bugs at least, don't you think?
Yeah,
I would
kind of.
guess so, but I have no.
There are also a couple questions in the chat.
Go ahead, Lance.
Well, I don't have them all up, but I just
Oh.
wanted to point them out so you guys can scroll down.
Okay, I see a few. I see one. Mary asks, the white-eyes diet in Newport, or states seems to be nectar and jelly rather than native seeds. So that could be again the ornamental trees and
Yeah.
Tori says they're mostly eating ficus, figs, and PV. Oh, and Kimball says that California Department of Food and Agriculture apparently successfully eradicated all of the Indian white-eyes in San Diego. 300 individuals.
Wow.
Wow.
I was way off. I thought it was
Double
just a
apart.
handful of birds, so that's really good to know.
Yeah. More comments from Merrill. They're all over the fruit trees in North Long Beach.
Yeah.
Pomegranates are popular.
Interesting. Interesting.
I have a.
I wonder if
I see a
that is
question from
during
Betty.
non-breeding season, and they go for insect for protein for the young.
Yeah.
Oh,
I see a question from Betty. If I scroll up, do you have info on the breeding cycle?
I don't have any info on the breeding cycle. Yeah, that's a good question, and they're pretty subtropical, as I mentioned, so I'm not sure it would be terribly different here than in their native range, but one of the hypotheses for why they're such a great speciator and why they've generated so many lineages is that they, as we mentioned, are like generalists. They'll eat almost anything, so they show up to an island and they can usually find something to eat, and that they have what seems to be, like, mostly anecdotally, one of the fastest breeding cycles of any bird, that they can pull off multiple clutches. And I believe there was evidence from specimens that were collected that within like 5 or 6 months of being born on some of these tropical islands, that they're reaching sexual maturity and able to start breeding themselves. So there's not a lot of hard evidence for it, but it's definitely one of the hypotheses that that's the reason have the characteristics that they do, is because of an extremely rapid breeding cycle.
interesting. Interesting.
Yeah. Kimball notes that there's a lot of photos of swinhoes with insects in their bills.
Oh, okay. Okay.
And photos of nests and fledglings. And breeding season seems to be mainly May to August, based on stuff in Macaulay Library and iNaturalist.
Cool.
Where people have photos of nests and things.
In the Q&A, Rosie asks, in areas where different species come in contact, do they hybridize? And if so, what do you think would happen to the Southern California population if 4 more white-eyes of different species were released here?
That is a great question. My answer is yes, because we have these birds we sequenced that were pretty confident are hybrids. I didn't include, there's a few more genetic analyses in the paper that sort of corroborate this as well. And if you look at where they came from, this hybrid comes from an area of overlap between Palpabrosis and Simplex. And then this hybrid down here is from, I hope I don't get this wrong. Is that Singapore? I believe it's Singapore.
Yes.
And whatever, it's a major metropolitan area in Southeast Asia. I'm sorry if I'm getting it wrong, but I have it outlined. It's kind of hard to see. And but I have it outlined by like 4 different species, because I think what I read happened is that originally Palpabrosis or one of the other species was native to this area, and then basically the urbanization and collection of the native white eyes for the pet trade was so intense that they were extirpated. But they now have white eyes again because of this released mixed flock of like 3 or 4 different species, basically exactly what you just described, because they're so popular as songbirds in Southeast Asia. So um, we sequenced, I think, only 2 birds from that locality, and one of them was a hybrid. So yeah,
Interesting.
I'm relatively confident that if you dump a bunch of different white eyes into the same place, that they will hybridize, which is also interesting from an evolutionary perspective.
Yeah, challenging to say the least. Wonderful. I think we're running out of questions.
Oh, I've got one.
Oh,
Great,
please go
go
ahead.
for it.
It's a big picture kind of question. So you mentioned that the speciation or the diversification is unusually rapid. Could you compare it with, say, the finches on the Galapagos? I mean, some of their evolution is very fast.
That's a great point. Yeah, I thought about framing it a bit that way actually, but I couldn't tell you off the top of my head how fast the Darwin's finches have radiated. The main difference that I wanted to highlight here, but I wasn't sure how it fit into the story, is basically like geographic versus adaptive radiations. That the white eyes are a total geographic radiation, where this single generalist white eye phenotype reaches tons of isolated islands and then becomes tons of isolated species. The Galapagos finches are an adaptive radiation, meaning that we think one ancestral finch or one or a few ancestral finches got to these islands, and there were all these open ecological niches. So then this one thing turned into the tree finch, the small tree finch, the woodpecker finch, the cactus finch. So it's just a different mode of diversification. And I would argue that adaptive radiations have been like almost overstudied, that everyone thinks adaptive radiations are so cool, which they are, but the majority of speciation that happens on Earth, evolutionary biologists agree, is allopatric speciation. It's things getting to a new geographic location or a population getting bisected by mountains or a river, and then the two independent geographically isolated things become 2 different species. So this is actually much more common, and I think we need more good study systems for understanding the dynamics of this allopatric process. And yeah, this was, one way I was going to frame it is everyone loves adaptive radiations, but actually geographic radiations are even more important for understanding the evolution of life on Earth. So thanks for teeing that up, Lance.
Interesting. Well, I think we've run dry, and Devon will give you a chance to hit the bathroom. Thank you very much for a wonderful, wonderful webinar tonight. We really, really appreciate it.
Yeah, it's fantastic. Very interesting. Definitely come back and give us another webinar sometime about when
Scrub
other
jays.
things, or about scrub jays or
Love to hear
or
about
anything
scrub
else you're
jays.
working on. I mean, yeah,
That sounds
fantastic.
great. Yeah, I really appreciate it. And yeah, I had a great time. It was yeah, a real treat to be able to present for y'all. So thank you.
Great.
All right.
Thank you very much.
Thank
And
you.
thank you everyone for tuning in. We will see you at our next webinar. Until then, good night, everyone. Thank you again. Take care.
Night.
Good night.